So this is the first lesson for lower sixth on the Beak 2 side, where we're going to cover the biochemistry, introductory biochemistry and the biochemistry of water. I'm hopefully going to do this within a reasonable time scale, which hopefully then will mean that the video doesn't drag on too much. So these are the objectives for the first lesson. So we're going to recap covalent bonding, what it is, what the definition is. We're then going to look at a new word, a new concept to you called electronegativity, and we'll define that and explain why it is important. We will then look at the factors that affect the electronegativity of various atoms. We will then look at the structure of a water molecule from a sort of chemical perspective, and finally link the structure of water to its biochemical properties, and then why that is useful in biology. Which is really important for many other topics. So this is quite hopefully a straightforward start, but the concepts are really, really key and come up many times throughout the A-level course. So let's look at objective number 1, which is looking at covalent bonding. We'll start with a definition of covalent bonding, which hopefully you remember from your GCSE chemistry, which is the electrostatic force of attraction, FOA, force of attraction, between a shared pair of electrons, E minus there I'm using just to represent an electron, between a shared pair of electrons and two oppositely charged nuclei, which may all just sound like words, and you maybe learned that for GCSE chemistry, but of course what we mean is this. So let's look at diatomic hydrogen here, and you can imagine that this diatomic hydrogen here is sharing this pair of electrons here. So both of these are electrons. The cross, let's say, it's from the hydrogen on the left, and the dot, it's from the hydrogen on the right. It's a shared pair. So essentially this electron comes from this atom, this electron comes from this atom. And we can represent this as a sort of like a overlap of atomic orbitals, kind of like this. So we can draw the hydrogen like that, draw the other one like this, and we can represent the two electrons in a sort of overlapped space between them. If you do chemistry, this is the overlap of atomic orbitals forming a molecular orbital, but don't worry too much about that. It is this pair of electrons here in yellow that is shared. And what you usually assume is what we've drawn here with this diatomic hydrogen, so this would be represented as H2, is that the electrons are equally shared between the two atoms. So if I get rid of my arrows here, we can imagine that the distance, which we kind of assume anyway here, and the distance here are equal. And that would be true for the case of diatomic hydrogen. Just as a quick aside here, looking for the numbers of electrons that are required, the easy way to work out the number of bonds, number of covalent bonds, that are formed between atoms is to remember the acronym 1, 2, 3, 4, HONC, H-O-N-C, HONC, kind of like a car horn. And the idea here is that this tells you how many covalent bonds each of these atoms want to form. So hydrogen here wants to form 1, oxygen wants to form 2, nitrogen wants to form 3, carbon wants to form 4. The reason is to gain a full outer shell. posh word for outer shell is known as the valence shell of electrons. So we'll use oxygen because it's an important example. If I draw oxygen in red here, I'll label it in the middle, so this is oxygen. If you were to go to the periodic table, which I will hopefully have here, don't worry about the electronegativity thing for now, but this is group 1, this is group 2, group 3, 4, 5, 6, 7, and then there is a group 8, of course, here, the noble gases that aren't shown for a reason we'll get to in a minute. Oxygen is sitting happy over here in group 6. That means it has 6 electrons in its outer shell, so to get to the full 8 that it wants to, it has to therefore gain 2 electrons to get to 8 in its outer shell. So if I go back to this, so it's in group 6. So I'm going to put 6 electrons in the outer shell that belong to oxygen. So let's do them as dots. So I'm going to put 1 here, 2 here, 3, 4, 5, 6. Don't forget to put them singly first, and then we start to pair up. So it's going to make 2 extra bonds. Okay, it will make 2 bonds, 2 covalent bonds are made to gain 2 extra electrons. So I'm going to show that here. So we're going to make a bond with an atom that has an electron to give, so maybe something like hydrogen here. So if I draw the overlap here, this would be hydrogen. And that will happen again. So if I just get rid of this, that will happen again over here. And therefore oxygen has formed 2 bonds, 2 covalent bonds, one here in yellow, the other one here. And it now has 8 electrons in total in the outer shell. So that's 1, 2, 3, 4, 5, 6, 7, 8. We have the full 8 outer electrons, so we are happy. Or oxygen is happy, metaphorically speaking. So you can repeat that for any of these atoms, and if you want the cheat sheet, 1, 2, 3, 4, honk tells you exactly how many covalent bonds these atoms need to form. If we look at this oxygen, what you will notice is, and it may not have been mentioned before now, but I'm going to mention it because it's important if I get rid of these ticks, one there, is that not all of the electrons in the outer shell, not all of these 8 electrons are used up in chemical bonding, in forming covalent bonds. 4 of them are, which I've highlighted in yellow, but if I highlight the ones in pink that aren't, it is these guys here. These are not used up in chemical bonding. So I'll write that here, not used up in a covalent bond. So, because they are sitting there by themselves, they're not being used up in a covalent bond, we do actually give them a special name, and we call them lone pairs. So in pink here, I'll draw the arrow down here, we call them lone pairs. So we've got one set of lone pairs here and another set of lone pairs. The importance of a lone pair we will get to in a minute, but essentially if you wanted the definition, it is a pair of electrons that are not used up in a covalent bond. They're not used for sharing because we don't need to. We've got a full octet, we've got a full shell, valence shell, outer shell of 8 electrons. In this case with oxygen by only forming 2 covalent bonds. Hopefully that makes sense. So that's the first objective ticked off. That's just a very quick, reasonably superficial recap of chemical bonding, but there you go, from hopefully GCSE chemistry. And we will now look at electronegativity, which might be slightly new to you. So we will start with a definition, which is the electrostatic force of attraction of an atom, technically the nucleus, which we'll get to, but in our case you can just say an atom, for a bonding pair of electrons. The electrostatic force of attraction of an atom in a covalent bond, specifically the nucleus of that atom, for a bonding pair of electrons. In simple terms, this essentially means how greedy, in inverted commas, is an atom for electrons. If you have a high electronegativity value, then you hog electrons. You are very greedy. So I'll write greedy here. You hog electrons. Essentially, you pull them towards yourself. And the opposite is true. If you have a low electronegativity value, then you will not be as greedy. You will be not as greedy. And therefore you don't hog electrons. You're sort of more happy to share electrons rather than try and keep them for yourself. Now, not all atoms have the same electronegativity value. The key ones that you need to know, so the key electronegatives that really do this well are fluorine, oxygen, and nitrogen. These are the ones you need to know. These are electronegative. We don't tend to come across fluorine very much, so don't worry too much about that one, but oxygen and nitrogen, we absolutely do come across in a lot of biological molecules, and therefore you need to know that they are very electronegative, and that will have an impact on function, on the chemical reactions that can happen, which you will see throughout the course. And of course, we've come across a molecule like this before, but let's just look at a single bond. So what we said is, if we have a covalent bond of diatomic hydrogen, the two electrons are sitting fairly uniformly in the middle. They're spending most of their time directly between the two atoms. That is because the atoms that make up diatomic hydrogen are the same. Seems an obvious point, but it's important. This means they have the same electronegativity as each other. And we'll look at the values in a minute, which means they exert the same pulling power, the same greediness for this shared pair of electrons. So the hydrogen on the left here is pulling this electron pair towards itself, and so is the other hydrogen on the right here, to the same degree. The force here, which I'll just represent as F, is exactly the same. These two equal each other, therefore the electrons spend most of their time in between the two hydrogen atoms. This changes when we change the atoms involved, so they're different. So let's have a look at a hydrogen-oxygen bond. So again, this is a covalent bond, and if we were going to put the electrons in, you might be fooled into thinking we'd put them there. But they are different atoms. We've got our hydrogen again, but we've got an oxygen here. And they therefore have different electronegativity values. And hopefully you remember this acronym up here, FON. Oxygen is one of the key electronegatives that you need to know. It's very, very greedy. Hydrogen actually isn't very greedy at all. It's sort of reasonably low electronegativity, especially compared to the likes of fluorine, oxygen, and nitrogen. What this means is that this shared pair of electrons is going to be pulled by the hydrogen over here, but it's also going to be pulled by the oxygen. And because oxygen here has a very high electronegativity value compared to hydrogen, so very high electronegativity, and hydrogen is relatively very low, that means the electron pair, the shared pair of electrons, is going to be pulled with greater force, which I'll represent by a fatter arrow, towards the oxygen, like so. Horrible arrow, but hopefully you get the point. Okay, oxygen has a lot more pulling power. It's very, very greedy for electrons. It's got a high electronegativity value, which means the attraction between the nucleus, don't forget, that's technically what we're referring to here. So if I draw that in, right, both of these have nuclei. So we've got protons in the hydrogen and protons in the oxygen. The protons in the oxygen have much more pulling power for electrons in a covalent bond than hydrogen. The reason for that we will get to in a minute. So that's the key point. This is all about electronegativity, hogging electrons, how greedy you are. And of course, what this means is if we're going to draw this accurately, if I get rid of these two here, we would actually put the electrons in this covalent bond, this single covalent bond, much closer to the oxygen. The electrons are going to be spending the majority of their time in the middle here in pink, closer to the oxygen atom than the hydrogen. So if I redraw this down here, if we draw the electrons in, I'll do them in pink, they're much closer to the oxygen. I'm slightly overdoing it here just to make the point, but they're very close to the oxygen. And hopefully you know from your GCSE chemistry that electrons are negatively charged. They're negative. What that means is if they're spending more time near the oxygen, the electron density map would look like this. Whereby at any given time, the electrons, probability-wise, are going to spend more time near the oxygen atom than the hydrogen. The important point here is because they're negative, we are more negative over here than we are near the hydrogen. This is less negative. Because the electrons that are negatively charged are spending more time near the oxygen than the hydrogen. It's not, just to be clear, it is not that they're spending less time, or no time rather, near the hydrogen. They are. It's just overall they're spending more time near the oxygen because it's got more pulling power, greater attraction for the bonding pair of electrons than the hydrogen does. I hope that is clear. I'm really emphasizing the point because it's so important in all of biochemistry. So, what this means is that actually this bond is not a neutral bond. It's actually something that we call polar. And how we show that the oxygen here has a little bit of negativity compared to the hydrogen is we use the Greek letter delta, lowercase. This means partial, or a sort of slight, a little bit. So if the electrons are spending more time near the oxygen, although we're not forming ions, we're not breaking this covalent bond here, the covalent bond remains, the electrons are spending more time near the oxygen, therefore the oxygen is slightly negative. It's more negative, slightly more negative than the hydrogen. We therefore represent this as the oxygen is delta negative, and because the negatively charged electrons are being pulled away from the hydrogen, more often than not, the hydrogen becomes delta a little bit positive. So when we draw an OH bond, doing it the other way around here, we can add our delta charges now. We can say that because oxygen is electronegative, it is going to be delta negative. The hydrogen, because it's its electron in this covalent bond, don't forget one of them belongs to hydrogen, let's say it's the cross, is being pulled away further than it wants to be. Therefore, the hydrogen is becoming a little bit delta positive. Because there is charge separation here in this bond, the electrons are spending more time near the oxygen than the hydrogen. And don't forget, this is what the electron density map would look like. We say that this bond is not a sort of neutral equal bond. This bond is a polar bond. We have two poles. We've sort of got a positive pole and a negative pole. But just to be clear, these are not ions. If ions were going to form in this bond, if I draw this again, if the electron from oxygen is a dot and the electron from hydrogen is a cross, what we would need to happen is that both of these electrons here get given to the oxygen. It pulls them off completely and breaks this covalent bond. What this would produce is this. It would produce an oxygen and a hydrogen completely separated. They're no longer overlapping their atomic orbitals. They're no longer sharing a pair of electrons because oxygen has ripped off hydrogen's electron completely from the covalent bond, essentially. So now both electrons, we've got oxygen's original electron here as a dot, but it's now gained the cross. It's gained hydrogen's electron that did exist over here on the hydrogen. And because oxygen is normally neutral and it has gained this extra electron, this now would be minus. So this would be an ion. We'd say it's O minus. Hydrogen has lost its electron, the cross here, the X. And if it's normally neutral and loses an electron, loses a negative charge completely, it becomes positively charged. And these would be ions. But I really want to make the point this does not happen. What happens is oxygen is a little bit, it's more electronegative than hydrogen and therefore can pull the electrons towards itself, but not enough to rip them off and form ions. Essentially, this doesn't happen because oxygen is not electronegative enough and hydrogen is not weak enough. Hydrogen's electronegativity is too strong to give it up completely. Essentially, there's not enough pulling power by the oxygen for the hydrogen to form ions. So what happens is the electrons are just pulled and the pulling power is not enough to rip them off completely from hydrogen, but they spend more time near the oxygen. And that leads to not ions, but a polar covalent bond, where we have a delta charge, delta negative on the oxygen, it's a little bit negative, and delta positive on the hydrogen. It's a little bit positive. So that's what electronegativity is, and we will now go to the next bullet point, which is the factors that affect it. So this is bullet point or learning objective number 3, which is the factors that affect electronegativity. Now, the attraction here is between a positively charged nucleus within an atom and electrons that are being used in a covalent bond. So let's put the electrons in here. And essentially, it doesn't matter what this atom is really for now. The key point I want to get across is that this positively charged nucleus, because we've got protons in here, don't forget, is going to be pulling the negatively charged electrons. It's going to be attracting them, essentially. So what's actually going to be happening is the electrons are going to be pulled in towards the positively charged nucleus to a certain degree. And it is this pulling power here in yellow that is represented by the electronegativity value. In other words, how greedy we are, this atom on the left. Hopefully, you can appreciate the following. This value is affected because of 3 things, 3 properties of the atoms that are involved. Number 1 is nuclear charge, which is determined predominantly by the number of protons. So you can imagine that if you have an atom here with 1 proton, the attraction for electrons in pink here in a covalent bond is going to be reasonable. But if we added a proton, so if we go for another atom here where we actually have 2 protons in the nucleus, where we've increased the effective nuclear charge by 50%, you can imagine that if we have our electrons in a covalent bond, the pulling power is going to be greater because we are more highly charged. There's a greater difference in charge between the protons, between the sort of atom on the left here, and the electrons that are used in this covalent bond. So they're going to be pulled closer towards the nucleus of the atom on the left. So that's nuclear charge. The next thing is something known as shielding. And shielding is important and has an effect because if we have, let's say, 2 atoms, they have the same, let's just say they've got 1 proton in the middle here, and we again have our covalent bond. Hopefully you can appreciate that the pulling power here is going to be reasonable, but if we increase energy level, in other words, we go to a different period within the periodic table, which I'll show you in a minute, we increase the number of electron shells. So let's just draw this in here. Imagine we get another atom here where actually we have 2 electron shells, where we have electrons existing all around, just to make the point, and we're forming a covalent bond here between these electrons in green. Again, hopefully you can appreciate that what is going to happen to the pulling power, the electrostatic force of attraction between the positively charged nucleus and the negative electrons in the covalent bond in green here, I'm highlighting in yellow. Hopefully you can appreciate that because we have an increased number of layers, right? It's kind of like an onion. We've got the inner layer here and we've got an outer layer. Therefore, there is more stuff in the way. there are more electron shells getting in the way of the positively charged nucleus in the middle here in green, and the electrons in the covalent bond in the outer shell in yellow, highlighted in yellow there. Therefore, there is weaker pulling power. There is less pulling power by the atomic nucleus, the proton, for the shared pair of electrons. So here, the electronegativity value of this atom, here, the electronegativity is smaller, is lower, is less than this one here that has less shells. It's only got one shell here, so there is no other barriers to the electrostatic force of attraction. So here, the electronegativity value will be greater. There is a third thing that affects this, but I'm not going to worry too much about it for now. Essentially, the atomic radius, which is kind of linked into shielding, but don't worry too much about that. I think I'm going to skip over that. Just as a sort of synoptic link, between the sciences, if you're doing A-level chemistry, this links to a topic that you do fairly early on called ionization energies. So when you come across this, hopefully you'll sort of remember this particular video, or certainly in the lessons and your biology notes that I've given you, and you can link to electronegativity as well. Okay, so that's electronegativity done, hopefully. The next thing is to look at the structure of a water molecule. So this is objective number 4. We're sort of getting there now. Everything's going to come together. So we're going to look at the structure of water. Hopefully you know that the formula for water is H2O, and therefore it looks like this. One oxygen and 2 hydrogens connected by covalent bonds. Don't forget oxygen here. Don't forget our 1, 2, 3, 4 honk. Oxygen forms 2 covalent bonds. We've got 1 here and 1 here. So oxygen has a full octet, a full outer shell of electrons. Let's put the electrons in. And of course, don't now put them in the middle, because the atoms are different. They will have different electronegativity values. And hopefully you appreciate that this oxygen here is definitely electronegative. So the electrons, both the hydrogen's electron that it's contributing to the covalent bond, and oxygen's, are going to be located far more towards the oxygen than the hydrogen. The electron density map would look something like this, where you've got greater electron density towards the oxygen, and they spend less time near the hydrogen. So we can add our deltas on. Oxygen here is delta negative. Our hydrogens are both delta positive. The other thing just to mention that we're going to add here, I'm just going to move this delta negative sort of over here, because oxygen has additional electrons in the outer shell. You always need to show all outer electrons when you draw dot cross diagrams. And don't forget, oxygen is in group 6, and it therefore has 6 valence electrons in the outer shell, and it needs 8 to complete its outer shell, to complete the octet. So we are missing 2, which I'm going to do in green here. So let's say oxygen's electrons are dots. So I'm going to put 2 here and 2 here. And hopefully you recall that these are called lone pairs. They exist on their own and do not contribute to covalent bonding because there is no need. They're paired up, although as you'll see later, that doesn't necessarily stop them from forming a covalent bond. But there is no need to form a bond because we have the required 8 electrons in the outer shell, which of course is the most stable electronic configuration. So don't forget the lone pairs. That might be new to you, depending on who you were taught by at the GCSE. It's not required really for GCSE biology. It is briefly mentioned, I think, for GCSE chemistry, but don't worry too much if you haven't covered it before. It is simply a pair of electrons that belong to an atom that are not used up in covalent bonding. So, the importance of this, hopefully you can appreciate now we know the charges. So let's draw 2 water molecules. Actually, let's why not? Let's draw 3. We can now begin to put our delta charges on. I'm not going to draw the electrons just anymore, just for ease, but we can draw our deltas in. So we're going to draw delta positives for our hydrogens, and we can put our delta negatives in for our oxygens. And actually, we're going to put our lone pairs in as well. So 2 lone pairs on our oxygens, like so. And hopefully what you can appreciate is because of the electronegativity value of oxygen, it is pulling the electrons in the covalent bonds with hydrogen towards itself, making the oxygen delta negative. But more importantly, it is making the hydrogens delta positive. And therefore, if you have a delta positive hydrogen that is close enough to another electronegative atom, and actually oxygen here has 2 lone pairs. Don't forget these lone pairs of electrons are negatively charged because electrons are negative. What this means is we can form a bond. I'm just going to change color here to a different color. Let's go for blue. Why not? What this means is if you have a slight positive charge on this hydrogen and it's close enough to an electronegative atom, such as, here we go, oxygen. Don't forget this is delta negative. What happens is, I don't know where that's gone actually, delta negative, there we go. What happens is you can get an between molecules, intermolecular force of attraction. And we will show this by a dotted line. We must use dotted lines because it's not a covalent bond. Please, please, please remember that. Intermolecular forces between molecules, inter means between molecules, not within, we must use dotted lines. So here we go. Our lone pair of electrons on that oxygen, the delta negative oxygen, is attracted to the delta positive hydrogen of a neighboring water molecule. And hopefully you can appreciate that the same thing can happen here. We've got another lone pair on here, and this can therefore attract to this hydrogen in the vicinity. And we can keep going. Of course, if we have another water molecule here, and we put our lone pairs in, our deltas, don't forget delta positive for the hydrogens, delta negative for the oxygen. This can also form intermolecular forces between the delta negative oxygen and the delta positive hydrogen. These intermolecular forces we give a special name. These intermolecular forces are called hydrogen bonds. Technically, they're an intermolecular force rather than a bond, but in the sort of traditional sense, but we can call them hydrogen bonds. And together via these hydrogen bonds. The definition of a hydrogen bond is when you have a delta positive hydrogen atom, it's the electrostatic force of attraction between a delta positive hydrogen atom. That's really important. It's called a hydrogen bond because you have a delta positive hydrogen atom and a neighboring electronegative atom. And of course, in biology, the key players here for our electronegatives are fluorine, oxygen, nitrogen. Specifically, oxygen, whoops, that's way too big. Oxygen and nitrogen here are the key players for hydrogen bonds. You need a delta positive hydrogen and typically an electronegative atom, either oxygen or nitrogen. They're the key players. Not the only ones, but the key players for A-level biology. The importance of these hydrogen bonds is as follows. This is an absolutely critical point, is that individually, they are incredibly weak. They are intermolecular forces. They're not covalent bonds. They're not even ionic bonds. They are sort of weak bonds formed from delta positives and delta negatives, little partial charges in covalent, polar covalent bonds. So important is that they're individually very weak. We can break all of these one by one, very easy to break this one, etc. But the importance of this in biology is that collectively, as a collective, they are exceptionally strong. Which means they take a lot of energy to break them apart when they're together, when there's lots of them. If there's only a few, then it doesn't take much. And you'll see why this is important when we get on to the properties of water. I'm just going to plug my laptop in because I think it's about to die, which wouldn't be good for anyone. There we go. I think I've saved it. I hope I have. Right. So that's the definition of a hydrogen bond. That's the structure of a water molecule now at A-level biology. And we've talked about delta charges and, importantly, lone pairs. And don't forget, technically, although you can just say that the hydrogen bond is between a delta positive I should put that here, actually. And of course, by electrostatic, we mean literally positives and negatives attract. That's what electrostatic means. But the electrostatic force of attraction is actually a product of the delta negative oxygen, the electronegative oxygen pulling electrons to form the delta positive hydrogen atoms, which is absolutely critical over here in our definition. So long as you're near an electronegative atom, then you will then form hydrogen bonds. And in this case, it is between the electrons in the outer shell of oxygen, the lone pairs, and the delta positive hydrogens. But in your exam, although I've drawn it like that, you are more than welcome, if you wish, to simply draw them like this, with your delta charges, and not bother about the lone pairs. And you can then simply draw your hydrogen bond like this. But don't forget, per oxygen atom, we can form 4 hydrogen bonds, technically. As you can see, well, I've not done it here, actually. As you can see here, if there was going to be another water molecule here, for example, and you had another bond like this water molecule here is forming 4 hydrogen bonds, which I'll highlight in red. 1, 2, 3, 4. Right, we are nearly done. So finally, number 5, this is the final bullet point. We need to relate all of this to the biological properties of water. And importantly for your exam, you must link this to the biological significance, importance. Don't just state the facts. This is not chemistry. Don't just state the chemical facts that underpin water as a molecule. You have to relate this to why we care. Why we care in biology, right? Why does this help us? Why does this help humans? Why does it help animals survive, essentially? So it's all about survival. Everything is about survival. It's a really important point, that. In fact, so much so, I'm going to highlight it. You will lose marks if you just simply state the biochemistry. You have to state why it's important. And that's what we're going to do now. So hopefully we're only sort of maybe 10 minutes or so, 15 minutes away from finishing. So the properties of water are as follows. We'll go with property number 1, which is that it is a universal solvent. And what we mean by that is it dissolves the majority of compounds. It is useful because it allows things to dissolve in it, right? We can dissolve solutes to form solutions. But the importance of this in biology, of course, is that this allows water, which by the way makes up the majority of you, it allows the water in you, which I'll just draw in blue here, the water in you to act as a transport medium. It allows cells, don't forget we've got a cytoplasm, right? You might have a nucleus here with your DNA in it. You've got sort of ribosomes dotted around, you've got mitochondrion, not to scale, but there you go. Most of the cytosol is actually water. Most of it is, or are, lots of water molecules. And it's important that you are able to dissolve substances, such as glucose, for example, because otherwise it wouldn't be able to be transported in your blood, technically your blood plasma. So in pink here, if I do blood in, what's happening? If I do blood in red, the red bit of your blood, going back to GCSE, hopefully you remember is your red blood cells, your erythrocytes, your RBCs. And the sort of watery substance that the red blood cells sit in is called the plasma. This contains a lot of water and allows things like glucose, amino acids, salts, etc., vitamins to be transported in your blood and then get into the cell cytoplasm via different mechanisms, so transporter proteins and that kind of thing. So there'll be a sort of carrier protein here, which allows them to get into the cell and then diffuse in solution to then go and carry out their function or be metabolized in the case of glucose. If you couldn't dissolve these things in the blood, then this couldn't happen. So it's really important, transport medium, because we can dissolve stuff, essentially, because water is the universal solvent. Why, from a biochemical point of view? Well, let's take a very simple example of dissolving. First rule is that like dissolves like. Polar things. If you are non-polar, then you can dissolve non-polar things. things like fats, for example. And because we've looked at the structure of water already, looks like this, don't forget, because we have our delta charges, delta negative, delta positives, these two bonds here are polar, so it is a polar molecule. Therefore, it can dissolve polar substances, things with sort of charge distribution. And the most extreme example of something that's polar, I hope you can appreciate, is not something with a delta charge, it's something with a formal charge, ions. These are the most polar things in existence, right? So something like sodium chloride, don't forget, is made up of a lattice of your positively charged sodiums interspersed with, and I'll try and do this like this, in a giant lattice structure interspersed with your negatively charged chloride ions. I'll just put negatives actually, like that. And what happens is this all gets broken up in water. I didn't expect that to delete. Oh well. And the ions dissociate to form individual sodium ions and chloride ions like that. The reason we can do this is as follows. And I'm going slow just because I really want to labour the point. We have a bucket full of water. I'll do water in a light colour here. The water molecules are in blue. And of course they look like this, and they have the delta charges. So let's do that. Delta negative, delta positive, delta positive, delta negative, delta positive, delta positive. There's lots of them. I'm just drawing two for now. When we dissolve our sodium chloride solid, our salt, and we put it into water, what happens is the sodium and the chloride ions dissociate. The reason is as follows. The sodium ion is positively charged, the chloride ion is negatively charged. Because these are charged, these are polar, they're the extreme of being polar, they're ions, they're charged, the water molecules can surround them. The water molecules actually break them up. So the water molecules break up the ionic lattice. And this is because they have delta charges, they're polar. So let's look at the sodium as an example. I'll just get rid of this water molecule for ease of drawing. What happens is these water molecules will surround sodium. And hopefully you can appreciate that what is going to interact. It's going to be the positively charged sodium ion is going to interact with which part of the water. Hopefully you can appreciate it is the negative bit, the delta negative oxygen. So water molecules actually surround the sodium like this. Because the delta negative oxygens are going to be interacting electrostatically, the electrostatic force of attraction, with the positively charged ion. True of chloride, it's the hydrogens that will interact with chloride. Because they're delta positive and chloride is delta negative. And the reason that things are able to dissolve is because it is energetically more favorable for all of these little intermolecular forces to form between the water molecules and their ions than it is to keep the ionic bonds in the solid together. This links to my theory, so I've called it the Doc-H theory of solvation. I don't know if it's mine or not. I've not seen anyone else use it. I've came up with it, I think. So For now, it's mine. Doc-H theory of solvation, which is that this is akin to or similar to a Scotch egg. It's a Scotch egg theory of solvation. In other words, when we look at this, when we've dissolved something, let's take the chloride, for example, it looks a bit like a Scotch egg. I mean, it doesn't, but the analogy is a Scotch egg, because a Scotch egg has the outer casing, the sort of bread, the breadcrumbs and the sausage meat, and in the center here, you have your egg. And the analogy here is that the center is the chloride ion, that's the egg, and the water that surrounds the chloride ion here, if I just color it in like that, is the bread, the sort of sausage meat casing. And we call this, the water surrounding the chloride, or the sodium, for example, a solvation shell. And I think the word shell is quite nice there because it links to the whole egg analogy. So water breaks up the ions, forming solvation shells around each ion because of the electronegativity of oxygen and the fact that water is therefore polar. Weak intermolecular forces, lots of them, can therefore form between all of the water molecules and their cognate ion, either the sodium or the chloride in this case. By the way, just to be clear, this holds true for any molecule that is polar. You don't have to be ionic. So long as you've got polar bits, polar part, you're polar enough, then you can dissolve in water. Things like fats that have no real electronegative atoms in them are not polar. There aren't any charged groups. Therefore, they do not dissolve in water. Okay. So that's kind of 1 and 2 together. 1 is that it's a solvent. 2 is that it's sort of transport medium. That's why it's important, but they're kind of linked together. So let's go for number 3. I think this is technically. Hopefully you've come across this equation before, which is that energy, or the sort of energy transfer, equals mc delta T. And this was in calorimetry when you did that at GCSE. We use this term, and you're not really told what it is or why it's important. It stands for specific heat capacity. In this case of water. And this is defined as, and you probably need to know this, the energy required, the heat energy required to raise the temperature of a known mass, typically a kilogram, but technically it could be anything, known mass of water by 1 degree C, 1 degree Celsius. The units are typically kilojoules per kilogram per degree C. You may see it as joules per kilogram per degree C as well. It depends what the number is, but typically you were given this. You were given somewhere in the region of 4.18, something like that, as your value when you did this last year for calculating energy changes when you were burning food. Why is this important? Well, the importance here links to the hydrogen bonds in water. The fact that you have many hydrogen bonds that are very strong collectively mean that this is true, that you require more energy than you would imagine to raise the temperature by 1 degree. Okay, it links to the hydrogen bonding. Don't forget, individually they're very weak, but collectively, when you've got millions of water molecules knocking around, they're very strong. Okay, so water has a very high specific heat capacity. But don't stop there. Why is it important? So we've said about why it's true and what it is. It is important because water acts as a temperature buffer. it resists change a lot of energy to change the temperature of water, to raise it by a degree Celsius. Therefore, it's important for survival of organisms. Things like fish in a pond. The temperature in their local environment is not going to change very much when the sun comes out in the morning. If water had a very low specific heat capacity, then when the sun came out for an hour or two, the pond would heat up, possibly too much. So we'd have maybe too much heat, and therefore the fishy would die. And that links to obviously enzyme activity and things from GCSE, but don't worry too much. Key thing, it's a temperature buffer, which means that it resists changes in sort of small fluctuations in temperature, which means that organisms can survive because the temperature in their environment remains sort of at a fairly set level. So protein, enzymes don't denature and all of that stuff. Okay, the last two are fairly quick. I don't know what's happened here. Ooh, I've got a rubber. Oh, there we go. Right, which number are we on? Number 4. Property number 4 is that heat of vaporization. What this means is that it takes a lot of energy to turn water from a not a solid, a liquid into a gas. In other words, evaporation. This is true, again, because of hydrogen bonding and the fact that collectively hydrogen bonds are very strong. So if I draw some in here, I'm not putting the delta negatives in just for ease at the moment. There we go. Collectively, if we have loads of hydrogen bonds, H bonds, it takes a lot of energy to break them all apart collectively to release each individual water molecule as an individual entity to become a gas. And don't forget particle diagrams, right? Particle diagram for a liquid is that you have some of them touching, some of them not. Like that. And to turn into a gas, to vaporize it, evaporate it, the particles need to be completely spread out, moving in random directions with high energy. So in this case, we've got some hydrogen bonds forming. And in this case, we have broken every single hydrogen bond. The hydrogen bonds don't exist. And we have therefore successfully converted from a liquid to a gas. And it takes a lot of energy to do that because hydrogen bonds are individually weak, but collectively very strong, and you have a lot in liquid water. Number 5, we're getting there, is that the solid form of water, so H2O solid, is actually less dense than the liquid, which is reasonably unusual because solids tend to be more dense because the particles are packed closer together. But the reason why when water molecules pack closer together when you decrease temperature means that more hydrogen bonds can form. So don't forget, if we're liquid, we might look like this as a particle diagram. And when we go for solid, they pack closer together in a fixed arrangement where they're not moving. It's less dense when we look at water. The answer, as you can probably guess, is hydrogen bonding. In the liquid, the water molecules have enough kinetic energy to move around. And essentially, the hydrogen bonds they form, let's say here, 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 and here, don't all form at once. Because these particles, these water molecules, have high kinetic energy, it means because they're moving around, individually these hydrogen bonds are weak, so they're going to break. So at any one point in time, this one here, because this molecule that I'm circling could be moving around quite fast, it could break off these hydrogen bonds and it could reform a hydrogen bond with a water molecule that's close by, but it's a different water molecule. This water molecule could then break off perhaps here, and it might move a bit closer towards the left-hand side here because it's got kinetic energy, high kinetic energy, and it could reform a hydrogen bond here, for example. It's all very sort of stochastic, and sort of the hydrogen bonds aren't fixed. They're kind of breaking and reforming all the time. When we end up like this solid here, when we decrease the kinetic energy enough, we decrease the temperature enough, the particles move around less frequently and less fast, less quickly. So what this means is the water molecules can pack closer together, and essentially every hydrogen bond that's possible is going to form. Like this. Because they're not moving around with enough kinetic energy to break the individual hydrogen bonds. We don't have enough kinetic energy, even though we don't need much. We don't have enough when we freeze water to break these hydrogen bonds. Therefore, they remain. And what you end up with is a lattice structure, a lattice of water molecules. And because we get this lattice, they are arranged at fixed distances from each other. Because every hydrogen bond is formed, this distance is fixed, this distance is fixed, so is this one, so is this one, so is this one, this one, this one. Which means essentially you end up with this. If the water molecules are circles here, you end up with water molecules connected to each other in a sort of lattice-y structure like this at fixed distances from each other. And that means that you've introduced spaces in green here. You've introduced a space here, 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 here. They can't pack close enough together. They pack closer together when you're in liquid form. This means that you are less dense because there are sort of think of it as sort of air gaps between the molecules, but the sort of gaps between the molecules, they pack slightly less close together because they are arranged in a fixed lattice structure, because they have less kinetic energy there, so more hydrogen, all possible hydrogen bonds form, and we don't have enough kinetic energy to break any of them individually. Therefore, the solid form of water is less dense than the liquid. But again, why do we care? Why is it important? You don't just get the marks for spouting this on an exam paper. You need to say why. Why is it important that ice floats? Well, if you imagine we have a lake, we've got our fish in the lake, many fish. When it gets to winter and we start to get solid ice forming, which I will do in a darker color. Let's say we start to get solid blocks of ice here forming like this. The ice, this is ice forming. The ice will then, because it is less dense than the liquid, move to the top of the water. And what you'll end up with is the top layer being ice. The liquid, or the water underneath, can therefore remain as a liquid. Because what the ice does at the top, which if I just highlight that in yellow, what the ice layer here does is it acts as an insulator. Kind of like an igloo. actually acts as an insulator from the cold, which is coming from outside of the lake here, and essentially it stops any of the liquid water underneath freezing, which means that the fish again don't die and they're happy. Fish survive. And you can then have your salmon en croûte for lunch. Again, always keep asking why. If you remember the American flag from my introduction lesson to the course, this is essentially how you get marks in the exam. It's not just for explaining the chemistry, it's for explaining why it matters. The final one, this is the last one, which I'm not going to spend a lot of time on. I'm just going to tell you because we cover the topic in a lot more detail later. There is the property of cohesion and adhesion. Cohesion is the attraction between water molecules. And adhesion is the attraction of a water molecule to another molecule that's not water. So attraction between water molecules, as you can see here, via hydrogen bonding, so here, is known as cohesion. You adhere to something when the water molecule is attracted to something else. So typically, you know, if it's in a glass sort of beaker, I guess, if you had a beaker of water like this, you would have cohesion between two water molecules here. That's cohesion. But the water molecules at the side of the glass, if we were to zoom in here, and I draw that again, the glass is made up of silicon dioxide, and the water molecules will be attracting to the side of the glass. Don't worry about how it's because silicon is polar. So the similar idea here, you get sort of hydrogen bonding occurring either between water molecules or to other molecules. And that is why, if you're interested, when you look in a test tube and you put water in it, you get a meniscus formed. Oops. And actually it kind of looks like this. So it's attracted to the glass at the sides, and it looks like that. I'm overemphasizing it here, but you get this because of adhesion. And this is both, these properties are both important, and I'm not going to go into the details now because there's a whole topic on it, for transport in plants, for transpiration. Specifically transpirational pull, which is the force, if you have a plant here, when water evaporates on a hot day through the stomata, any water in the stem, because of cohesion and adhesion, is going to be pulled up the stem into the leaf, because don't forget the water molecules here that are sort of breaking free were connected to each other, right, via hydrogen bonding. So therefore, if you move one of them up the stem, then the rest will follow in a chain. Right, hopefully, we've done a lot there. Hopefully that makes sense. So that's we've covered introductory biochemistry, the definition of electronegativity, the factors that affect it, the structure of water, and then the properties of water. Very common question is to relate properties of water based on the structure to biological function and importance. The only thing just to mention with regards to the periodic table is electronegativity values are shown here. You'd never need to learn them, but hopefully this just emphasizes the point I said earlier, which is as you go across the periodic table from left to right here, electronegativity increases. So 3.04 to 3.44 to 3.98. Fluorine is the most electronegative. We don't count group 8, the noble gases, because they're noble. They are inert. So they don't participate in chemical bonding. And the reason that this is, hopefully you can appreciate, is because within a period, within an energy level here, this is an entire energy level, the shielding remains the same, but we're adding a proton. each time. Therefore, the nuclear charge increases, the shielding stays the same, therefore the attraction for the electron by the proton in the nucleus is greater. And as we go up the group, electronegativity increases because of the shielding changing. We are reducing shielding. So hydrogen only has 1 energy level, lithium has 2, sodium has 3. We're increasing the distance from the positively charged nucleus, the proton, to any electrons in the outer shell used in bonding. So hopefully that makes sense. We have covered these things. Any questions, send me an email or see me outside of lessons.